Uh, yeah, thanks. Bye. We now move on to the next, the next session on regulatory aspects. The session is actually broken up
to AMR, related to AMR and the antimicrobials, though the processes for regulatory and clinical design and development are generally similar because it's driven by basic regulations, basic design, and ICH requirements and stuff like that. I will obviously each one of these, aspects of design and regulations, we'll take it on a very broad level, at a general level. We will not get into specifics because every specific topic is a topic in itself, which will take hours to get into details. But what I'm trying to convey today is what are the broad requirements, the basic fundamentals of regulations that will be associated in any clinical development program, and also then give a very broad idea about some of the devices and the diagnostics, because a lot of people are into that space, which is a part of this whole development program. And I know in the last few years, we have been having a lot of challenge in India, especially related to device regulations and device laws. And there is a relative, not a confusion, I would say, but not clarity in terms of what needs to be done at what point in time. So I will try to address that in a broad level. And though my slides might be mixed together, but we'll try to address each and every point as we go through the presentation. Before we get into the presentation, I also want to make a statement here that most of my slides are not my own slides, because I have taken these from different presentations done by different people. Some of them have been mixed, but the whole purpose of that is for purely educational purposes. So that's where some of these slides might have been seen by people and presentations made by people, but the whole purpose here is to explain and to give an understanding of how the processes and the systems work. All right, so let me try to, is my presentation visible?
Yes, we can see it.
Okay. So I'm trying to put all the regulatory and clinical considerations for anti-infective clinical development programs, though these are not only specific to clinical development and of anti-infectives, but even in general. But here we will talk more about specifically to this. So a little bit of background on this. See, regulatory requirements, whenever we design a program, whenever we have, let's say we have reached a clinical stage, so the first and foremost thing, we want to have toxicology done, and now we want to design a study, a phase 1, phase 2, phase 3, the whole program, the clinical development plan that we design for us. So the plan itself in terms of development is started based on what are the regulatory requirements, because ultimate goal is that my product should get to the market. And there is a certain steps, there are certain steps involved in that whole process. And just to give you a little bit of background, I will talk about, just let me go. So regulatory requirements across. Now, whenever we develop a program, anybody who is a startup and has reached to a clinical stage level now, they would want that this program be in a particular country. And then the focus is to go global, because you want, the regulations are driven primarily by what has been done in the US, by the US FDA, the EMA, most regulatory agencies, the PMDA, and then we look at from our own country's perspective. And so the whole objective should be that I should not be doing multiple things for different regulations. So we try to kind of converge what has been happening. Now, as a part of that, the regulatory agencies also have kind of tried to come together so that, the requirements of the regulatory agencies also seem to be kind of matching, so that it's not that any company or anybody who is coming with a product is not developing different programs for different regulatory agencies, so that it is uniform and there is not a lot of money spent on designing and developing that program. So some of those steps, I will just give a little bit of background on this. So as we said, regulatory requirements across countries and authorities are different, and several steps by regulators to converge on these requirements to reduce the impact of development has been there has been a big initiative on this. Now, especially when we talk about antimicrobial resistance, we know we are having this as a problem, a big issue globally, and in India even bigger. So we say that India is antimicrobial resistance is the capital. Like I used to hear about diabetes capital of the world, and now we hear about antimicrobial resistance capital of the world. So it's a big issue. So regulators also have understood that there is a need, and how can we converge some of these regulatory requirements. So a lot of effort on aligning these requirements has been happening in the past, and some of these, like might have heard about the Task Force on Antimicrobial Resistance, they have been seeking this convergence in multiple meetings in the last 4 to 5 years. A lot of things have happened, and they have typically focused on specific areas which are our concern as far as antimicrobial resistance is concerned. In acute, I listed some of those: acute bacterial skin and structure infections, community-acquired pneumonias, and complicated urinary tract infections, and the other hospital-acquired and ventilator-associated bacterial pneumonias, because these are where we are finding the most of the resistance is developing, the organisms that cause all these infections. So there has been an initiative which has been happening over the last few years. And some of the common indications, in addition to what I'm suggesting here, have also been pursued in terms of these regulations. One of the important area for discussion has been the clinical requirements for products addressing unmet medical needs, which is the AMR, which we're talking about. So we have been trying to find out, okay, what are those clinical requirements? What are those endpoint requirements that are needed for developing these programs when we design these studies? And some of the recommendations do vary between FDA and EMA. For example, in the FDA, they recommend a mortality endpoint for such infections which are serious infections. And the EMA and PMDA recommends clinical cure endpoints. So these recommendations are different. So which means some level of innovation around this has to be brought in terms of regulatory acceptance of data, and then the design of the trials will be accordingly defined by the people who are developing these products. Sometimes a well-conducted single trial in a specific type of infection due to an organism may be good enough, or with an appropriate comparator, with the support of a PK/PD analysis, for example, which should be adequate for accepting a data package. So regulations, so that's where the regulations are trying to go towards. And as a result of these combined efforts by multiple agencies, so they publish their own guidelines or guidances, as we call them. They are more of guidances, though they are not laws, but generally these are the general understanding of what is required to be designed, to be developed, and how the data packages should look like. So they give those guidances, and they are legally not bound, but that's what generally is followed and practiced even by all the regulatory agencies, and in fact referred to even when they come for inspections and audits. So if we go by the history of what has been happening in the antibacterial drug development in the 60s and the 80s. Nandita, one point, I'm just getting this, can I just get okay, yeah, thanks, sure. Thanks. No, I was getting some, okay. Oh. So patients with a variety of infections at different body sites were enrolled in the same trial. And objectives to demonstrate comparable point estimates to active control for clinical cure for each of the different infection types. So this is something what used to happen in the very early on, when we had not many, the issues of resistance were not there. We were trying to find out treatments for infections and infectious diseases. So this is something what used to happen in the past. Indications were based on subsets of body sites of infection from within the trials, and less specific indications, example, lower respiratory tract infection and skin infections. These are very broad, common kind of infections which were being, for which antibiotics used to be, developed and approved and then used. As we moved into the 90s and the 2000s, there was a move towards more site-specific trials. So natural history of the disease of that particular infection may differ. Endpoints and treatments and the duration would be different. Drug efficacy may differ at different sites of infection, and the dosing regimens might be different for different sites as well. So that's how the 1992 IDSA guidelines came in. And then the FDA, along with that, the points to consider document was developed, the clinical development and labeling of anti-infective drug products. So some of these are the progression that has happened in the whole development of regulations and the development of programs for anti-infectives. And then there was a recognition of the aforementioned differences in these documents represented an advance in clinical trial design. So there has been this change happening over several years, and that's how the transition has been going on. Now, during the 2000s, especially in 2006 and around, there was a significant turmoil in the field of scientific questions which were raised about the non-efficiency, the trials. Basically, when we try to do non-superiority or non-inferiority trials, so that becomes a little challenging, and questions are being asked. In one regulatory agency, if it is approved, then the other regulatory agency would question it and would not accept it. So it became very difficult. So considerable effort and stakeholder participation were in designing scientifically sound trials and evidence-based trials with margin justifications, trial conducted to common indications. All this went on, and in 2012, there was a focus on unmet need, particularly to treat gram-negative infections as these infections became more and resistances started coming in. So there was a focus towards all of this. And this streamlined the drug development programs were pursued, and often single trial per indication, smaller safety database, between 300 to 500 patients would be acceptable by most regulatory agencies. Though at that time also, sometimes we had to do more than 1-2 trials sometimes to address the needs of one regulatory agency versus the other. But there was an initiative happening towards how the different regulatory agencies would come together, because it was the same companies trying to market their products all over the place. So in the upcoming years, continued focus on unmet need programs, including difficult to study indications, continued, and the development of non-traditional therapies also got into the action. Now, of late, in the recent approvals, if we look at the types of data packages that have been generally standard and accepted, have been basically on standard indications, as we said, community-acquired UTI, complicated UTIs, and community-acquired pneumonias. Then there was a system where they said 2 trials per indication, or at least 1 trial per indication, should be required for getting into to get your regulatory approvals. Then there was a limited use indication, a single trial with supportive evidence-based, phase 2 study, in vitro study and animal model as a support to the whole data packages. Now, it would again depend, because the label would typically drive, and we'll talk about the label as we move forward. The label would define what exactly your trial design and what kind of trials you would do in terms of getting into, to develop your program, basically. And then the small data packages, single trial, well-defined and limited population of patients given unmet needs. Some flexibility in the benefit risk considerations were also considered by regulators. So it has been, over these years, this has been off late recent approvals. But if I recall, in the last few years, I think last 4 to 5 years, we haven't had any, anti-infective approved, significant one which would address most of our needs. Okay. So why are we here today? So we are in 2022 now. While we have made progress, it appears that we are at a critical juncture in antibacterial drug development. And I think AMR basically is being talked about all around the world. And multiple nations, multiple consortiums have come up to address this kind of an issue. And it brings a big challenge to the development community, the drug developers. First of all, not much investment is going into this area over several years. And that's something that you will probably discuss as we move on during the sessions. But there has been a criticism regarding the clinical utility of some of these recently approved products and the registrational trials conducted to support their approval. While there is an unmet need for some difficult to study indications like osteomyelitis, prosthetic joint infections, we need to work and ensure to map out the needs and potential solutions. While labeling is an important component of the discussion, addressing the scientific and feasibility issue is the key. So when we talk about designing a program, designing about a product, so when we have reached there, so I think we have to start with an end in the mind. What is my label going to be? What is the product that I am coming up with? What is it going to address? So we have to work backwards and try to understand and design our programs in that fashion, because that's how the regulators will accept it. And based on that regulatory requirement, that's the way I'm going to design my studies accordingly. Some of the criticisms regarding recent registration trials, I will just get a little bit to get you some perspective on this. There was this clinical condition studied in complicated UTI. Such trials are feasible and demonstrate the efficacy of the product at a body site, which is basically the urinary tract, which provides reasonable safety information in a population with fewer confounding factors and allows for a step up to a more difficult to study condition. So we need to balance the realities of drug development with the desire to study difficult indication populations. So a lot of times what we happen, now we might, when we design a program, so from a scientific point of view or from a molecular perspective, we would like to get all the answers. But when we look at the patient population, we may not be able to get all the answers in that single trial. And that is where we keep on multiplying programs, and it's very difficult depending on the population that we are looking at. And when we design our programs, it's not necessary that we should look at answering all the questions that we might have, to address within that particular study, because a lot of investment has already gone in. The idea typically is that can we get a regulatory approval based on a basic concern or the basic question, the hypothesis that I'm asking for, or the basic target disease that I'm targeting, and then look at subsequent trials and add on indications and look at other aspects and get to that, because then by that time you are able to make some money by selling the product and you can add on. So sometimes all of these are extremely critical parameters in terms of your study design and in terms of the questions and the hypotheses that you design when you're trying to build your programs. Sometimes lack of data in patients with infections due to resistant organisms. Conducting a randomized controlled trial in patients with infections due to resistant organisms can be challenging, because it's very difficult to design such a kind of a trial, especially when we are doing the randomized controlled trial, because randomized controlled trials are the norm of accepting from a regulatory perspective, because those are more scientifically driven. So recently conducted trials were difficult to interpret as they were descriptive trials without any pre-specified hypothesis testing. So some of these basically are criticisms which have been raised during recent approvals and recent data that was submitted to the regulatory agencies and were approved. And when the same company is submitting this data to one authority, and when they go to the other authority, they have a difficult problem there. So as new therapies become available, the resistant phenotype of interest also can change, and that is also one has to really consider, especially with the resistance that is happening. Am I getting some sound? Sorry,
Yeah, requesting others to please stay muted, because we are getting some background sound. So please do stay muted. Sorry.
So the potential trial designs that we look at for most of these antibacterial programs or antimicrobial programs are finding of a superiority over the best available therapy. So that's a typical standard. So we have an available therapy and I have a new product and I want to compare it head to head and actually find out the superiority of this particular product compared to the other product. And then we can also do an enriched trial population in a non-inferiority trial if an appropriate comparator is chosen. Now it depends on what infection, what disease we're taking, what are the available antibiotics for that particular infection at that point in time. One can design, a study which is either a superiority study or a non-inferiority trial. Both are acceptable, but typically, in general, if I have to make a statement, the superiority trial is always the best to do. But depending on what disease and what antibiotic options we have and what we want to compare it with, depending on what the product looks like and its function, we can design those kinds of studies as well. Mostly non-inferiority trials, new products only, and non-inferior to the existing therapies. Yeah, there was a lot of criticism regarding this, as I mentioned earlier on. You might do a very well-conducted non-inferiority trial and are interpretable, and they might provide useful information on the efficacy of a product. But, as I said, it might be acceptable because, risk profiles and the way the regulators look at it can vary, can be different. And that might be a problem. So demonstrating superiority to currently available therapies is difficult, which is why people try to get onto a non-inferiority kind of a study saying that, okay, my product is as good as XYZ, which is available for this particular indication. So that is the reason why people try to get into the non-inferiority, because all other challenges of population and the disease, the epidemiology and everything else can be challenging sometimes. Most drugs we use today were in fact approved based on finding of non-inferiority. Through superiority is of course helpful if shown. So that's what I was saying. So mostly it has happened that way. And a lack of very few patients enrolled in this happened in the US FDA actually. Because US FDA typically sees, okay, what is the number of, because they typically approve products based on what they find within their population and their addressing, because that's what every country tries to do. And if so, for multiple reasons, people do go out. One is there may not be many infections happening, especially in the US area, and then getting patients into the trials might be difficult. So general guidance is typically, though it's not written anywhere, is that you should more than half of your patients, if you're doing for a US registration, more than half of your patients should be from the US. And there have been situations where they do, though it's an unwritten thing, but that's expected. And sometimes we land up into those situations as well, where we have patients which are being recruited elsewhere and not for the US. So you have to think that, especially US companies who are doing it, or if I'm developing a product in India and I'm targeting the US market, I have to make sure that I'm doing a lot in the US, because that's where my regulatory approval would be required. Patients with comorbidities and more severe disease often excluded. Now, assessment of PK in patients with hepatic renal impairment early in drug development can help limit exclusionary criteria. When we get into the design of the studies where we look at the protocols, when we define exclusion and inclusion criteria. So sometimes we do also look at the PK in patients who have hepatic and renal impairments. And then the flexibility in inclusion and exclusion criteria were ensuring patient safety. So these are some of the points that one designs in the study, but these were the points that were kind of questioned when some of these products were have been registered in the US or in the EMA, or for that matter, Japan as well. So basically, when we talk about clinical trials, well-designed comparative clinical trials teach us important and unexpected lessons. So if we have a well-designed clinical trial, we get a lot of lessons, and we use those lessons to develop further programs as we get it. So, for example, binding of drug to surfactant interactions identified after failed CABP trial, which was daptomycin. Higher mortality and lower cure rates in a VABP trial, which was digoxin and doripenem. So there is altered pharmacokinetics in sick patients, augmented renal clearance issues, differences in lung penetration between animals and humans. So when you actually get into deeper and try to see those differences in the data, so these become questionable, that whether these are the products coming in are the right ones or not. So the importance of body site of infection, efficacy of drugs seen in one indication but not in others. So these are some of the points. So the whole reason why I have been talking about these are very important aspects when we design our studies. And some of these aspects we need to consider. While we may not be able to solve every single point, as I was mentioning, we may not be able to get all the answers at the same time, but it's always important to be aware of these points, that these are the areas that we need to look at, especially how the regulators have reviewed the data in the past, what have been the questions and the concerns that the regulators have raised in terms of trial designs, in terms of outcomes, so on and so forth. These are some of the examples. I will not get into the details of what happened in these. These were the findings in these clinical trials, unexpected clinically important findings in well-controlled clinical trials. So for different products like daptomycin, doripenem, and so on and so forth. So these are important aspects that we need to really look at and consider when we design our studies, when we design our trials. As I said, label is very, very critical because that's what you want ultimately on the product. Once you get through the registration, once you get through the trials, what will be the indication on my label? What is that I'm looking at? As I said, that is how we start. When a product is thought of, you develop a draft label, the SPC as we call it. What is it that I want on my label at the end of? So two key considerations for a label as labeling, really ensuring consistency, including information in labeling based on sound scientific evidence is helpful to all stakeholders, providers, payers, and patients. Because ultimately, whenever the insurance agencies and when the product comes to the market, it is the label that will drive everything else. The usage, the prescription patterns, the insurance agency will only support what is on the label. So that is very, very crucial. And our regulatory, our trial design should be addressing that primary question. That is that we want to make it on the label, that this antibiotic is to be used for this particular types of patients, even if it is a subset of patients where it is indicated, because based on the results of the trial, we have to be very, very specific and clear on that label. And as we get to the programs, the label can change depending on what outcomes we get of the study. Sometimes you draw a label as a very broad label, but when we get to the data and we find a subset analysis and say that, okay, it can be good in patients of this particular subset who have X, Y, and Z, then the label will be modified accordingly. So, but the plan of a label has to be started right up front. Specific requirements on content and format of labeling for human prescription drug and biological products and clinical study section. These are some of the general things which a label should have for drug products other than biological products. Any clinical study that is discussed in prescription drug labeling that relates to an indication. So these are descriptive things, what you have on the label, the dosage and administration section, the indications, contraindications, stuff like that, and all the warnings and everything. So it has to be driven by that. Indications include the organisms for which adequate clinical data were available in the trials, for example, and information regarding limited population, if applicable, what I just mentioned, what specific type of subpopulation is applicable. The microbiology section also in terms of the, sometimes labels even talk about the MICs and what has been seen, because those are detailed labels. And based on that, the in vitro activity has been defined in animal model and first and second line list of organisms. And then the clinical studies section describes the adequate and well-controlled trials that support the indication. So when we talk about label, it's not just the package insert, which is very small, but a little bit of detailed, as I said, the SPC, but not full SPC, but at least most of the important aspects of what the product has gone through. Same development program can result in different product labeling across different agencies, as I was saying, particularly for indications in unmet need areas. Again, because of differences in the regulations and the way it is approved, it is also important that labeling recommendations be based on sound scientific evidence so that it's helpful and clear to all stakeholders. But what happens mainly is that, the risk benefit assessments of antibacterial products, though, are fairly similar, but there are differences how the risk benefit assessment is done by different regulatory stakeholders, the advisory boards who sit down. Like for India, we have our SEC who define it that way. So basically, that's very, very important there. You might find some differences in that, in the judgment on that risks and benefits leading to different regulatory decisions. But that doesn't happen to only anti-infectives, it happens to any other product for that matter as well. So some regulatory flexibility, as I said initially, is important, but we have to be sure that the scientific underpinning of clinical trials are not compromised, because just we want to make sure that we get uniformity across. I would stop here for a minute. I think if Nandita, I was just wondering if we take questions even in 2 sessions, it may be okay. In between we wait for a break, we can take 2 breaks, like for questions. Yes. To address some of the questions, it might be easier.
Would you like to take some of the questions now?
Yes, why not?
Okay, so there's one question from Bhakti, which says, what are the regulatory and clinical trial pathways for new drugs to be used on compassionate grounds?
See, compassionate use is post-regulatory, actually. If you're running a program, I'll give you a general example here. If you're running a program for, like, say, an unmet medical need, and that program is running, so there are requirements. I will try to answer it in 2 ways. Now, we are running a study, and it is going on and is doing well. And there are certain patients, group of patients who respond to these, and it's an unmet medical need who respond to these. Now, the trial is over. What happens to these patients who have done well? So there is a requirement a lot of times that, even in India, that can we, these patients who have responded, a compassionate use program, and the drug should be available to them, post trials, because they have responded. And so you run a compassionate use program for them. And that's one of the requirements in India, that if a trial, which is patients who are responding to this treatment, access to that medication should be provided to those patients. Now, it cannot be given like a trial, so you have to run it as a compassionate use program, and they will be available. That's one way of doing it. The other part is that product, even if, for example, you have a product which is new and is not available here. So companies do run compassionate use programs. Now, there are a lot of, for these patients who are given, they typically will not pay for it, or they may pay for, if it's a part of trial, they may not pay for it. But if they have to pay for it, it is the company that bears the cost, and they will see all associated testing that is required for that is a routine process, is a routine standard of care. They might only provide the drug. However, from a regulatory point of view, if it is an unapproved product, all other product responsibilities that the company has in terms of follow-up of a marketed product may not be applicable here when the trial is in an unapproved condition. So we may not get into the details of those at this point in time, but that is the only way. So there is no program that, oh, I am developing a product, now I want to run a trial which is only compassionate use. No, it cannot be like that. It doesn't work like that. So you have developed a product to a certain point, and then if there are patients, a subgroup patients who actually respond, you want to continue on them, though your product is still in development and it may not have been into the market. So that's how compassionate programs are run. For products which are in development, you can run compassionate use programs post-marketing, which will be like, which the companies do run a lot of times, but for multiple reasons. I mean, we may not get into those reasons, but in that, if it is in the market, the responsibilities of regulations and the market for that product will lie with that company at that point.
Thank you. There's another question from Dr. Anand Gopakumar. What are the regulatory and clinical trial pathways for synthetic peptide new drugs?
Well, see, the pathways for any new drug, for that matter, whatever the source be, whether it is, there will be a difference between biologics, there will be a difference between synthetic products or chemical products. Per se, there is no difference. There will be a difference in these two approaches of regulations in terms of what a biological product or a similar biological will undergo versus what a chemical or a synthetic product will go. That is one part. So there is no difference in, that's the only difference, but you know, it is the same thing, trial design, toxicology, and then you get into the phase 1 and then into the phase 2, you might do a lot of characterization in biologics and so on and so forth. But the second part of it will be, what is this peptide doing? What is that mechanism? What it is? Does it affect the genes? Does it affect anything at the genetic level? So then there will be other additional regulations associated with it. Certain approvals have to be taken. But in terms of designing your studies, it will be the same, but the regulatory pathway might change depending on what requirements you are using. For biologics, for example, you have to go to RCGM and you have to go to the biological. If we talk globally, it goes to the as a new biological entity. In chemicals, it will go as a new chemical entity. And the pathways for that might be a little different. In India, when we talk about from an Indian context, you have to go to RCGM, you have to get clearance from there. If it is anything to do with with the genes, you will have to go to the stem cell committees, or you will have to go to the genetic. There's a new committee on gene trials, so you have to go to them. Otherwise, the DCGI will be the main focus point for your approval process, which will be there. So it will not depend on typically on only whether it's a peptide or non-peptide. It depends on what it does and if it is a synthetic versus a biological product.
Another question, are clinical trials needed for fungal and or plant extract in combating gut microbiome dysbiosis?
If you're trying to, any claim that we are trying to make on a, if you're, see, there are two ways. One is that you want to make a prescription drug. You have to do validation of whatever you are testing, whatever you are claiming. So any claim that you make on your product, be it antifungal, be it anything, or a nutraceutical or anything, if you're making a claim, you have to do a validation. Now, the study design and the type of studies might vary depending on what you are trying to achieve, or what your label wants to do, or what you're trying to claim. That may differ. But validation studies definitely are required. Clinical trials are required.
Another one, very useful, sir. What kind of drug mostly used in gut-brain connection for neurodevelopment disorders?
I don't think I'm capable to answer this question.
Yeah, probably out of the scope of this thing as well.
Right. No, even if it was out of scope, but I don't think I'm capable to answer the question.
Okay. And yeah, so Uma, maybe you can ask somebody else. Bhakti would like to thank you for your answer. There's another question, which I think you will be covering probably after this session. Could you tell us the regulatory pathway for biologics?
Okay, so all right. So we are talking about, I will talk about from an Indian context. So typically for biologics, once you have the biology, I'm talking about, is it a new biological entity, or it's a similar biologic? Now, if you are having a new biological entity, it will have to go through all the, I'm assuming that all the toxicology, all the characterization, all the lab, all that has been done. Okay. And the moment you reach to the clinic, I will spend more time on that. The moment you reach to the clinic, you are ready for, and assuming that all the toxicology and everything has been done in the clinic, you design a study. And so the process is you make an application. You have to go through the RCGM. Okay, so RCGM will look at your biological product. At the same time, once RCGM clears it, then you can go for a clinical trial application. You can have two routes, because if it's an Indian product developed here, you can go through the new drug application process straight away as an IND submission to the DCGI. So you make a full development plan and submit that what you are going to do, and start with the phase 1 study, and then go on to the next phases depending on how your response to the study 1 and study 2 comes in as you move on. So it goes under, like you in the FDA, you go under a US IND, you make it under one program, you keep on doing one study and another program and keep on submitting the data and move on to the next one. So the same process will happen here. Now, when you make to the DCGI the application, so there is a process there that there will be, I won't get into this, what are the documents and other things that you can always find out. You go there, there will be a committee sitting in who will, scientific committee, the SECs as we call them, subject expert committees, they will review it and they will ask you to change or ask you to modify the protocol or accept the protocol that you are designing and accept it, and you will go for that study. And you will design that study. Once you have designed and run that study, you will submit the data to them if it is a phase 1. Then based on that study, you are going to the next program. You will have to present the data and they will allow you to go to the next phase and then to the next. That's how it works. And in between, there are a lot of other programs that you have to do in terms of pharmacovigilance, in terms of safety management plans and other things, which you will submit as a part of your submission that you would have done. So that is the regulatory pathway. And once all that is done, you will have to go for the marketing authorization to the similar committee, for which you have to get a lot of manufacturing information, and which is quite humongous, quite a lot, registration of your site and stuff like that, all of that for the manufacturing part. But the clinical trial until you reach to the market, this is the process for regulations. And then, the manufacturing and other licenses you will have to take. All that is very well drafted, defined. If you look at the clinical trial rules, it very clearly defines that. And then you go by the Drugs and Cosmetic Act, and where it tells you what next, in terms of manufacturing and marketing authorizations you require. This is basically the simpler and short summary of what the process looks like.
Thank you. And then Dr. Anand and Ajesh have thanked you for your response. There's another question from Arun. Just a moment. Just come to that. There are a lot of startups offering genetic tests or microbiome tests without CE marking. CE IVDs. How are they regulated?
Well, okay. See, I think a lot of during COVID, during the pandemic, we had a lot of emergency authorizations for a lot of things, and because the situation was like that. See, normally, if you're going to market a product which is considered under device, okay, which is considered under a device, the CE marking or an equivalent is necessary. So I think as we go on, we will talk about what the device regulations look like. Maybe it will get a little bit of addressal there. But generally, a CE marking or a standardization is required if you come to the market. So that is required. But yes, if there is an Indian product, there are certain quality standards that are minimally required. CE marking, typically they look at products which are coming from outside, and that is the objective of everyone, but it may not necessarily be available at the first time itself. So there are certain what you call relaxations in that sense that are given, and that's probably one of the reasons why you see that happening, that they may not have CE marking right up front. Because if you have to go to the US, if you go to the EMA or elsewhere, you won't be, you will need to have a CE marking anyway.
Anuj has a question. Anuj has raised his hand. Anuj, if you can unmute and ask your question, please.
Am I audible, ma'am?
Yeah.
Yes, yes.
You can
Yeah.
even switch on your video if it's possible.
Okay. Sorry for that, because I am right now traveling, so it's tough. So
Okay, no worries. Please
That's
go ahead
okay.
with your question.
yeah, it's a nice explanation and all, exciting session till now. So I want to know that if there, because the drug has to be passed through phases of the clinical trial, so will it be possible to recall the drug at final stage if there is any mishappening? And I want to know that there is, because it's all about the regulation and the
Yes.
trial. So what is the fate of the patients which we have the already treated with
study.
these trial drug and all that, if that
Correct.
drug is mishappened in the final stage?
See, any study can be stopped at any point in time, which is phase 1, phase 2, or phase 3. Okay, because phase 3s are registration studies which are large, so a lot of money and things have gone into it from a developer's perspective. But patient safety is prime. Now, it depends on for what reason that trial has been stopped. And typically it would be if you find, a lot of trials, especially big ones, large studies, there are data safety monitoring boards which figure out whether there is logic in continuing the study. So the study could be because of safety reasons or sometimes because of efficacy reasons that you don't find. Sometimes you don't reach to the, and when you do an interim analysis, you don't find a statistical significance which would approach, like, for example, in the near future, we don't see much happening. There won't be any power that you will prove. So it is not, even when you know the outcome of the study, it is not ethical to even continue patients on the study because you're not going to do anything, because that's equally important. Second, that is one side from a positive side. Positive meaning that there is no harm to the patients because of the drug. Now, if the drug is not giving a lot of side effects and there are problems with the drug, then obviously it will have to be stopped because of safety reasons. So 2 reasons. Now, the question you asked is what happens to the patients who are doing? If for harm reason, it will be stopped. If for it will be stopped anyway, so no patient will have any problem. The patients will have the study will have to be stopped. The patients will have to be communicated that the study is being stopped, which is a part of the informed consent right from the beginning of the study, because you never know, because at the end of the day, it's an experiment, it's a trial. So it might have to be stopped anywhere in between. And so if for positive reasons, if we still find that certain patients were responding to this drug, and if the drug is available, but we don't expect, there can be a consideration that though the trial overall might not have a response, there might be a possibility that the regulators can consider as a compassionate use, but generally that does not happen, to be very clear, because if the drug is to be withdrawn or stopped, the trial has to be stopped for one of these 2 reasons. So there is no point, giving the drug to the patient, so that the trial will stop because it's not been a positive trial.
Yeah,
There's one more
actually, sorry.
Yeah, go
May
ahead,
I?
Anuj.
Yeah, this question arises because this in this COVID vaccine trials and all that, even after 2 to 3 phases of trials, we have the WHO or something like that, Indian agencies have given the authority to use it as an emergency use drugs.
Correct.
So the final trials are going on. So what if there is any mishappening, all that? So that's why I am asking this question, because
Correct.
a lot of peoples are already vaccinated, and what if we are, because till now we don't have the final concrete results with respect to these vaccine. So
Vaccine,
that's why I have
yes.
concern. So.
No, that's correct, because see, all these, because the situation was such that we did not have an option. So whatever little data we had at that point in time, the regulators came forward for lack of nothing. They gave an emergency use authorization to these products. Now, when we go into, suppose for a hypothetical situation, and that we are looking at the results of the trials at the end, and we don't find them to be of statistical significance or not. So the product might be stopped and withdrawn, but there is nothing wrong as long as there was a safety issue. See, that's where the difference is. If there is a safety issue, if there is a harm to the patients on a significant number, then it would be completely stopped. So whatever has happened based on the data that was available till that time. When the statistical sample and the population becomes larger, you might get to know more about the drug and some of the other issues that might be associated with the drug. But that was the risk that everybody around the world on this, they did that at that time.
Yeah, as you have rightly mentioned now that there are a lot of factors which are the responsible for the stability and all that. So this should be taken into consideration, and the subsequent passage of time, it will be finally, we can say with authority that yeah, our drug is very good and all that. So thank you so much.
Correct.
Yeah, thanks, Anush. Thanks, Dr. Kher. Two more questions, and then we'll just maybe take a 10-minute
We'll take
break.
two more. Yeah, two more
Yeah,
questions.
we'll just
and then
take
we'll
these last two.
Okay.
So Karthi Shanmugam is asking how to proceed with clinical trials in India of an already approved or investigational drug for its new clinical or therapeutic application.
So I think there are two different questions. One is already approved is one, because if it's already approved, or the other point is for an investigational drug. So the pathway will be different, because already approved drug, you are looking for a new indication, correct? So it will become a phase 3B kind of a study in that case. For an investigational new drug, the process is the same. You start with phase 1, phase 2, and phase 3 for an investigational drug. But if you're doing an already existing drug, which is in the market, and you want to look at for a new indication, for example, you are looking at indication A, now you want to include indication B into it, and because there's a basis for that. So because most of the safety data is already available, because you're using that same drug it's already available in the market, then in that case, you will have to do one single trial to compare a head-on-head versus what you are looking at, which indication you're looking at, and see that if it works in that indication. But for an investigational new drug, you have to go through the whole phase 1, phase 2, phase 3. For an existing drug, it's only a phase 3B, a new indication drug, and add-on.
Thank you. The last question. Sir, if I develop a polyherbal formulation against a disease, do I need to go through a clinical trial, provided that we have significant preclinical, that is in vitro, ex vivo, and in vivo studies data?
See, every product, whatever be the source, will have all its data. There is always a lab data, which is in vivo, in vitro, and then you might do toxicology and everything. So all that will be an indicator of what happens at the lab level. But what happens in the clinic is very different. So sometimes we get to conclusions based on what we have in our laboratory, and even up to toxicology, to in even human, even in primates. That does not necessarily mean that it will reflect the same thing when it comes to human clinical trials. So we have to see data, you have to validate, because you're giving with a prescription product. See, nutraceuticals and add-on for vitamins and all, it's very different. Because here you are treating an indication, a disease, for which you have to prove that it works better or as good as the standard of care, what is there. So it has to undergo a validation, it has to undergo a trial, a study, which will prove what we are trying to hypothesis, based on which we are coming to, which we have proven at a lab level, but we want to see that it happens in clinic as well.
Yeah, okay, thank you. So he says thanks very much, sir, for the explanation. So I suggest that we'll take a 10-minute break, maybe reconvene at about 12. It's 12:15 now, almost 12:15. Yeah. Maybe by 12:25.
Okay.
Or so. Have
Sure.
a quick cup of coffee or something, and then
yes,
we can resume
sure.
talk.
Sure, certainly.
Before that, can we just take a quick group picture so that
Yeah,
we
sure.
capture everybody, whoever, please switch on your videos, as many people as you can.
Yeah.
And yeah, if request if you can stop sharing your screen.
Yeah, I will
so
just
we can
stop
have
sharing.
everybody.
Yeah.
Yeah. Meghna, you'll take it, or should I take it?
Nandita, my connection is really bad.
Okay, I'll take it.
Yeah.
Sure, okay. I'm giving another 5 seconds for people to switch on their cameras, and then we have multiple screens. We've got a good participation, so please. One more screen to go, and then we can. Yeah, thank you. Let's all come back at 12:15 for the second session.
12:25, I guess.
12:25, 12:28 should be okay.
Yeah. Okay. Thanks.
Okay, thank you. Thanks. You can stay connected, all of you.
Yeah.
We'll just
Can
come
stay
back.
connected. Okay. Let me share the screen. All right. Now, since devices and diagnostics, basically diagnostics is a part of this whole spectrum of clinical research. So let's talk quickly about, we may not get deeper into details because I also want to give you a broad perspective subsequently about a clinical trial design and what types we have and what are the changes that are happening now in terms of developing programs. But since devices is a part of it, let's quickly go through what the device regulations are in our country. So as you would see, all medical devices in India to be regarded as drugs, the medical device amendment rules, which came in 2020. So as per that, I won't get into the details, but before October 1, all presently unregulated medical devices will have to be registered by respective importers or manufacturers with the Drugs Controller General of India. However, those medical devices which are already regulated or have been notified to be regulated are exempted from the requirement of registration. So which is 37 categories. We won't talk about which are those 37 ones, but and before August 11 of 2022, importers or manufacturers and the distributors, wholesalers and retailers of presently unregulated, which are class A, which are very low risk, and class B, low medium risk devices sold in India will have to compulsorily obtain a license. And then before August 11, 2023, importers and manufacturers, distributors, wholesalers, everybody else will have to compulsorily obtain a license for the class C and class D devices. So that this is the device law as of today. So the new definition which was there until February 2020 earlier on, the government has regulated the 35. So the definition is all devices, and this is very important, and everybody should understand what will fall under a device. So which means even if you are using the computer systems to analyze data, human data, that will also fall under a category of device. So all devices, including an instrument, an apparatus, appliance, implant, material, or other article, whether used alone or in combination, including a software or an accessory, intended by its manufacturer to be used specially for human beings or animals, which does not achieve the primary intended action in or on human body or animals by any pharmacological or immunological or metabolic means, but which may assist in its intended function by such means for one or more of the specific purposes of, number one, which any device. So this is a very broad and encompassing anything that we do related to human data, which will have any influence on diagnosis, prevention, monitoring, treatment, or elevation of any disease or disorder. Diagnosis, monitoring, treatment, elevation, or assistance for any injury or disability. Investigation, replacement, or modification or support of anatomy or of a physiological process, supporting or sustaining life, disinfection of medical devices, control of conception. So when we talk about devices, so a lot of currently we are using AI and ML now into a lot of medical and healthcare related outcomes. So anything out of that will fall under that category and will have to undergo the same regulatory process. This is just what I wanted to communicate and what a device means, because the understanding is device means anything where I am doing anything interventional, which is perceived that way. But anything, even a computer system or an algorithm used, is a device. Okay. The above new definition is intended to cover all medical devices as per technical discussion that preceded the notification of the above definition. Now, I will not get into the detail. This is the new definition. And requirement of registration, the manufacturers or importers of newly modified notified medical devices will be required to compulsorily register their medical devices with the Drugs Controller General India before October 2021, which was what we read. The DCG will start accepting applications and blah, blah, blah after that. So this is basically till October 2021, which is past. So everything now that has to be registered, accepting class 3 and class 4, which we talked about till 2023. Now, what is the process for device regulations? In order to obtain registration for medical devices, the importers and manufacturers of these devices have to be certified as compliant with ISO 13485 medical devices, the quality management systems. And this is what I was talking when somebody raised this question about CMARC, registration and CMARC. So ISO 13485 is absolutely essential and required for any device. So that has to be done, and that's a quality management process. So all importers or manufacturers or sellers of notified medical devices must obtain a license from the appropriate licensing authority before undertaking any commerce in notified medical devices. A license is issued only after a quality check. The license holder's business premise is subject to periodic inspection, and the license holder is also required to maintain detailed records of the sale purchase undertaken in relation to this. So these are the laws. Why, most of you, I think the participants here are still as incubatees or are developing devices, but with an end in mind, they might not be necessarily ready to market or do anything. But this is what has to be kept in mind while we are designing and developing such devices or such products. So the approval process, as we said, class B, C, and D IVDs, in vitro devices, require an in-country performance testing through, when we say in-country, it has to be done here in the country, performance testing through the National Institute of Biologics or an accredited lab. Class D IVDs require performance testing through the National Institute of Biologics. Class B and C IVDs require performance testing through an accredited Indian lab. And then the compilation of a device application, which is called a MD14 form. The manufacturing facility information has to be provided. There are much more detailed ones. I have put the main ones. Device technical information has to be there, which includes date and everything else, and what has been done, what is the shelf life, what will go wrong, right? Everything has to be done. ISO 13485 certificate, the instructions for use, testing results and clinical data, if applicable, depending on what we talked about, BCD, so which has to be done. And then if there is any clinical performance to be done separately, that also should be done. And for importers who are getting, because sometimes they use, a lot of times predicate devices are also used. So you try to kind of see that this similar device is applicable, is already marketed in ABC country. So you try to support it with this documentation. Or if an importer is just using the device here in India, and they just have to get an approval of the US, EU, Australia, Canada, or Japan. So this is, again, it's the same thing what I mentioned. A certificate of compliance with ISO 13485 devices has to be procured. Broadly speaking, ISO 13485 requires creation, documentation, and implementation of a quality management system, which is to be supplemented by an independent audit from time to time. So when you are getting to any ISO certification, obviously you have to have a quality management system in place, based on which you will get this. And that has to be updated and regularly audited internally, and will be auditable externally as such. Okay, this is all I wanted to talk about the device laws, what are currently there. But in terms of performance testing, in terms of validation testing, the designing of devices and trials is almost similar. But yes, we are not using products, but sometimes your products through a device, so the device is also tested. But the design generally is uniform. But yes, you don't get into toxicology and other stuff of devices. You just get into a validation study of a device, and then actually, based on that outcome, you can go for registration of your device based on, and also compiling all other requirements that we just talked about. So a few points about designing trials. We talked about so one thing which we know now is that we have to run clinical studies, we have to follow the regulations, and we have to design our studies based on the endpoints that we want. And I just wanted to add a few more points to that based on my understanding, so that there is clarity on what all needs to be done. Again, here I will not get into complete details. So what are the core elements of a clinical study you have designed, whether it's a phase 1, phase 2, phase 3, whatever we're talking about? So there has to be a research question. What are we trying to answer? What is our hypothesis? So that's very important, because as we talked about label with an end in mind, you start with the label. What is it that I'm trying to prove? So you have to design your research question based on that. And that's where we talk about what is the hypothesis. So some of these elements are, what is the core design? What is the basic design? We'll talk about it as we go on. Who will be the participants? Who are the target population for this particular product, for this particular study? Where are they located? What types of participants? Are they in-house participants? I mean, in-house meaning, are they admitted participants, or are they going to be in OPD? So all these parameters will make a big impact on your design and the operational part of how you conduct and execute this study. How is that recruitment going to happen? If I'm talking about study participants, are there participants? If I'm doing a study in sepsis, for example, where am I going to get these patients? How is the recruitment going to affect? How quickly I will get, because that's how you will define how long your study is going to be, what cost that study is going to take. All of these are very, very important. Similarly, then, when we are doing a study, am I going to mask the study or blinding the study? Because when you are comparing two groups, treatment groups, so how are you going to assign treatments to these two groups? If you're doing a product, an antimicrobial agent, let's say, for example, head to head between antibiotic A versus B, how is that? Are you going to blind? Will people know? Because you have to remove bias as well. Otherwise, I will know who is taking what. So my evaluation criteria, my clinical evaluation criteria, at least, if not bacteriological, will have to be looked at. And how am I going to capture this data? How am I going to do the data analysis? And based on how I capture this data, what is my statistical sample size? How am I going to interpret the results? Because ultimately, based on that interpretation, I'm going to design my next study, or based on that result, I'm going to make my submission to the regulators to get this study approved. So these are some of the basic elements of a clinical study. These are basic elements. So as we said, the research question is extremely critical in design of a trial. We cannot have a trial where we don't have the research question. So are we assessing efficacy of an intervention? Obviously, safety I'm taking as a given. Are we assessing the effectiveness of an intervention? So I will give you a difference in the efficacy and effectiveness. Efficacy, typically when we do a phase 2 or phase 3 trial, we are doing it under controlled conditions where we are testing treatment 1 versus treatment 2. And when I do the effectiveness, I typically, the intervention is there, I'm adding on other uncontrolled conditions and see that is it still effectiveness. So typically when we do real world data collection, we look at the effectiveness of an intervention as well. And when we are doing head-to-head comparisons, we are doing basic efficacy of an intervention. Now, trials typically, when we talk about a superiority or a non-inferiority trial. So superiority trials is where my objective is to demonstrate that my therapy, which is my product, N, is superior to the standard of care in terms of outcomes. So if a particular drug is used for this particular infection, I have a product which I feel, based on whatever data I have so far, is going to be superior to this study. So your sample size determination will be based on that. The power of the study will be based on that, because you have to prove that this is superior. Because typically you would see what is the difference between the two standard of care and the normal. And based on that, you will see how good or better your product is. You will design that study. In an equivalence and non-inferiority trial, especially when we do large scale trials, I am only trying to prove that my product is as good. So it's another product for the same indication that I'm coming with, but it is as good as and not worse than the standard care. So which means I'm giving another option to the patient and to provide them this therapy as well. In the other one, superiority, I am proving. So the sample sizes here would be different. So the difference between the two arms would be different in terms of sample size and in terms of the design of the study, the outcomes that we're looking at. So standard common designs are a parallel and a crossover therapy. This can happen for any type of trial. So you're trying a new therapy and you are doing the comparison. So you get two sets of arms, two sets of groups of patients, patient arm A and arm B. So in one arm, you assign them new therapy, and another arm, you give them the comparator therapy, which is the standard of care, for example. And now how do you assign these patients to this? You do a randomization, which is a blinded kind of trial, which is supposed to be scientifically sound and more acceptable by regulators, and where you are actually doing a randomization allocation of patients. So where you are doing a randomization based on allocation through a statistical system, and a computer generated allocation is done. So you don't know what patient is getting. And in this, again, we have single blind versus double blind. Double blind means the patient and the investigator or the treatment doctor both are, nobody knows who is getting what. They know there are two treatments, but who is getting what is not, so that there is no bias in evaluation. Okay, a single blind is that the patient doesn't know, because its patient is not aware of the therapy, but the doctor is aware of what he is giving to another one. So double blind, randomized, controlled trials are what is ideal for approval by the regulators, ideal. But that does not mean that it cannot be, but depending on, as I said, depending on what product, what trial we are looking at, and what we are trying to achieve as an endpoint. The trials can also be crossover therapies. So sometimes what happens is you want to test both products and to for both sides, sets of arms. Like, well, for example, you have a parallel, you have two arms, and for a certain period of time, you give the new therapy, and then you give the comparator therapy. After a certain point in time, you cross over, so that other group gets the comparator, and the one that had got the new therapy gets the comparator, and the one which has got gets the other one. So that's called the crossover therapy. Sometimes you, in between the crossover, you might give a washout period also, because you want to remove, depending on what product we are talking about, or sometimes you can equally move to the next one. So this is another design, well acceptable design, and it reduces the the differences between the two groups as well. The variabilities of the two group subject variabilities also get addressed in this kind of a design. I also want to tell you now, see, all the trials that we are talking about have been, we know that how a trial is done. Trials are done at hospitals, clinical trial centers, where you run these patients, you get these patients, and the patients come to the site, they visit the doctor, and all that recording is done there, lab tests are done. Periodically, the patient will come, the drug is distributed there, or the device is allocated there. Physicians consult them, the nurse and site staff is there, and the patient related data is collected there and there is a consent. After consenting the patient, it happens. These are the traditional trials that are 100% site dependent. Now, before the COVID came in, see, we are using now technologies in trials. So this has been going on for some time. Over a period of several last 10 years, there has been changes in getting into the electronic modes of, designing, even recording data. And so all that has been happening over the period of time. Now with current technologies coming in with computer systems, the trials are becoming more and more remote. Some of the activities, not necessarily all, but the trend is moving towards getting into virtual trials. So what does that mean? So, and how does it help? The basic purpose of those is because when we run big studies, it costs a lot of money and time. So time in product development is equal to money. So how can we get answers faster and quicker? Can we use technology to hasten our whole process of development? Now, obviously, everything cannot happen suddenly, and you cannot change. Certain things are not under your control all the time, so that can be done remotely. Because see, if a patient has to visit and you have to diagnose, the doctor has to see the patient. So some of it does happen, but there are a lot of other elements which can be converted into electronic mode. So as we are moving, pandemic has definitely helped us in this process actually. And for example, the meeting that we are doing today, probably we wouldn't have thought this 3 years back. So it has to be face to face. Even government meetings, everything has moved on to virtual mode. So similarly, the trials are also trying to get into similar mode. But yes, it depends on what product, what requirements of the design are there, which are necessary to be done remotely versus what can be done on the site. So this slide shows what is a typical 100% site dependent trial. The next one is, we call as hybrid trials, where there is a mix of both, off-site activities as well as on-site activities. So patient check-ins are happening, routine tests are conducted, drug distribution is happening, device allocation or shipment is also happening, physician consultation is happening, combination of virtual and on-site consulting. So you do get onto the combination of on-site and virtual. If you remember, before COVID came in, there was a law passed in virtual because virtual consultations with doctors was not allowed in India. And because of COVID, they allowed that. And that also helped us in the patient trials as well, because you could consult patients remotely. Though previously in our studies, we used to do telephonic consultation on follow-ups, but not on active treatments when you had to give therapies. Nurse and site staff assistants. So what started happening is, so combination of mobile nursing, telemedicine, and on-site nursing. So you're doing both on-site nursing. So if I have a trial, well, I have to give let's say three doses intravenously at the site, I'm doing it. After that, I have to only follow up and remotely check and collect some samples. So the mobile nursing can go and collect the samples at patient's workplace, at patient's home, wherever they want. And the doctor can consult through, video consulting to that. Similarly, data collections. For example, we do patient diaries to the patients. So the patients would fill in those diaries and would come back to the site and give those diaries. So they can do that remotely, giving them a tab or a mobile phone on which they can do the data. Now, when we design these data softwares and all, there are certain controls and conditions which are required in terms of security and privacy. All that is considered, all that is taken care of. But this is how this dual mode has started working in terms of hybrid trials, because still we may not be able to deliver. But if I'm using a new drug, a new chemical entity or a new biological entity, I can't do that at home. I will have to because I need to watch for safety. physically because it's a new drug. We don't know what's going to happen. So that way you cannot completely make it 100% virtual. But in that case, if I was using an already marketed product and I'm looking at real world outcomes of a study, I can do everything virtually. Even if I'm using an oral medication, I give my dose to the patient and say that, come on, I will talk to you after 1 month. You keep on taking the dose and samples and everything else. Is there, I think, some disturbance, some noise coming in?
Yeah, kindly stay on mute, please. I think by mistake.
Yeah.
Yeah, sorry about this.
That's okay. So the virtual trials, which I was just talking about, what is the change there? So you trials where there is no site dependence, but meta-site exists. There is a site existing for one particular hospital, but everything is done remotely. So what you'd use there is utilize telehealth and telemedicine. Patients do not visit the sites. They don't need to visit the sites. And mobile healthcare providers become your allies. So you collaborate with them. Like we have, samples being collected here, and you can get deliveries done to their of reports online. You can get even drugs delivered at their houses, okay, and devices at their houses. Virtual physician consultation, whether it is audio, video, text. Virtual in-home nurse assistants. I mean, in COVID, we did that. Happened quite to a great extent. In home care treatments happened, and that can happen in trials as well. Patient-related data is collected virtually on mobile healthcare providers, added, and telehealth or mobile informed consent. Even the informed consent is done. That is that kind of, we have been doing in surveys, for example, monkey survey, we take consenting and we do that. A similar version can be done on mobile. So you are running trials much faster, easier, with full control of data in a remote way. So these are 100% virtual trials. But yes, it depends on what products. For a new chemical entity where you need to actually follow up and look at safety patterns, you can do that. Now, in oncology also you can do that. I'm going away from antibiotics. In oncology, you might do that. First few doses you can give at the site, and if the patient is doing okay, you can even consider locally in that area only. They don't need to come to the site. Get it done at home. A chemotherapy at home also, or any other therapy for that matter can be done. So, but it would all depend on what product, what endpoints we're looking at, and certain points. Hybrid is already happening. A lot of things have become electronic. So we are in that mode right now, but moving towards virtual trials. And there have been a few which have been 100% virtual. So to summarize, over the last decade, we have made significant progress with development of antibacterial agents. New safe and effective therapies are available to patients. Though we have the bigger challenge of antimicrobial resistance. And as we said, a lot of initiatives are being done to address that. It's important to learn from our experiences and continue to refine our approaches to address patient needs specifically. Some considerations to encourage as we move forward is need to identify the types of infection and the patients in whom there is an unmet need. Novel study designs or endpoints that are scientifically sound, and improve clinical trial infrastructure.
Kindly unmute, kindly mute yourselves. Whoever is on unmute, please take a 5 seconds to check and mute yourselves.
And improve the clinical trial infrastructure, establish clinical trial networks, because that helps in terms of like what I was explaining just now. It helps to get your program run faster and finish the program quicker, so your product is ready and available to patients faster. Need to identify barriers and stimulate investigator interest in participating and enrolling in clinical trials for anti-infective products. Unfortunately, over the last several years, there has not been much development in the anti-infective area. And so interests of investigators has also kind of come down. But if we have really interesting opportunities coming up, I think definitely products are needed, the way resistance is going on, we definitely need to do more trials and generate that interest in development of anti-infective products. So I stop here. I think I'm trying to address whatever best could be within the short span of time. We can't get into deeper details, but just to give you an overview of how designing and regulations together are important for any clinical development program. So thank you. So open for questions, please.
Any questions? Please put them in the chat or just unmute yourself and ask the questions.
Here I have to submit only abstract containing introduction methodology.
Okay, all except will be divided under oral or poster presentation. So we don't want to do this.
See, if you go for this innovation, either you will be selected as one of the innovation presenting things. They have a separate room for that. Or you might be asked to present as poster or oral. Any three.
He's talking about poster, na? But you already have poster. But if we'll go through
Is this a question for us? Or?
So is it, I'm just wondering, so is it a question or what is it?
Nidya?
Only published. Not poster or something.
Here poster or paper only this is.
I think it's
This is what this is.
I think this is it's a side conversation,
Can you please,
I guess.
yeah, she's.
Hello, can I raise question?
Yes, please identify yourself and raise the question.
Yes, I am Pramod Stefan from Bihar, and I innovated one substance by juice, and I tested it in CDRI Lucknow, but till now there is no clinical trial and other things. No, I written many organizations and the central government, and I attend the National Drug Discovery Hackathon. I got certificate, but not work done in my innovative substance. Please advise me for the development of my innovative substance.
So let me try and if I can address that. So what did you do at CDRI Lucknow? What was tested there?
I made a drug, homeopathic drug, bile juice and rectified spirit, and then it is tested. I went CDRI and I told them for tested, and they tested but they not found any harmful things.
No, harmful is one, but if you have to use this, since it is an homeopathic drug, as I understand, you will have to approach Ayush, and because I'm not sure what all testing has been done, there is a process at Ayush. You have to go to that ministry, which is a part of.
I went. I went. Ghaziabad for Ayush testing. No. But they told me, they written me that this substance have no any pharmacopoeia, so we can't test it.
No, see, for homeopathic, you don't have to have pharmacopoeia. You don't have. I don't know where you have gone, but it has nothing to do with the pharmacopoeia. So if it is a natural product or a homeopathic product, you have to go to the Ayush ministry. You check there, because I won't be able to answer everything, but I don't know what it is all about. So the information that you are sharing is only preliminary, because if you have done the basic lab testing, then you need to go into the next phase.
excuse me, I went, we can make a homeopathic medicine, Unani medicine, Ayurvedic
Yes.
medicine, and allopathic medicine. We can use this substance in all pathy. I went Ayush ministry and all ministries since 2008, but nothing good done till now.
I'm sure they would have found out something. They would have asked you, because if your data and documentation is enough to go for an approval, because if it is a homeopathic product, then they would have told you. You should ask them what all is needed, what is required. I don't know what your data is, but you have to check and ask them what is the data, what is required for me in order to get this product to market. That should be the question. By writing letters, nothing will happen.
Yeah, but I want clinical trial, no, by
That's
some
right.
organization.
So if you have to do that, you can do that. If they have required and asked that you do this kind of a design, this kind of a trial, it can be done.
Actually, I have no any facility for the clinical trial, no.
Yeah, so Dr.
So, I
Pramod,
can't do.
yeah, so
Yeah.
I think if you find out from them that you need to do a clinical trial, there are many CROs
We can do it.
in
Yes.
India who can do this, do these trials as a service, on a pay for
No,
fee for service.
I am not satisfied because
No,
I
so
went
what you can do
CDRI,
is you can possibly write to us at that.
And government is also making a hackathon, a drug discovery hackathon, and I returned to a CSIR, but I didn't get any proper reply.
Yeah, Dr. Pramod, this we may not be able to address that particular issue of why you didn't get a response. Once you get a response, we may be able to guide you as to what you can do further. So if that once you get a response, you can.
I will send you my details.
Sorry.
I shall send you my details regarding my innovation.
Yeah, please do send it,
Please,
and
yeah.
also please send us after you get a response from them. So in both context of both, we can, if it is possible, we can guide you.
Yeah,
We'll just
correct.
go to the next question. Approval for drugs versus nutraceuticals.
Okay. So see, drugs, we have been very clear. Nutraceuticals basically are add-ons. They are not treatments necessarily. Now, if you are going to get, see, for nutraceuticals, typically it depends on what particular indication you want. It's not a treatment. If it's an add-on as a supplement, nutraceuticals typically are supplements. So you can go to FSSAI, get an approval, and you can use the nutraceutical. But if you want to get a prescription on it, then you will have to design a validation study for what you are targeting it against. So it will be like any other drug then. So if it is just as a supplement, as a nutraceutical supplement, you can go to FSSAI and get an approval for that based on obviously all the manufacturing, blah, 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 all the details have to be compliant with. But if you want to actually get it on a prescription kind of a thing, then you will have to do some clinical validation studies to prove your claim. It may not be as stringent and as controlled as we do typically in this. If you have got an FSSAI, so safety wise, there is no problem. Then you can run a clinical program on that.
Don't see any other questions.
Great.
So I think, yeah, with this, we come to the end of the
Thank
morning
you.
session. Dr. Kher, thank you so much. I think this is really a very complex issue, and you've really sort of simplified it and laid it out in a very simple and easy to understand, and provided all the the explanations wherever and also the questions where they came up. So thanks a lot. And
Thank you.
Yeah, if there's
Thanks
any
a lot.
further questions, or
Any
if there's
further questions, please
we
do
will,
send it to
we'll send it to you,
CCAMP,
we'll-
and please, I can be of help. Yes,
Yeah,
certainly.
sure.
Thanks, everyone. Thank you very
Thanks
much.
a lot.
Thank you.
And we will resume at 2 o'clock. We'll take a break for lunch. Come back at 2 for the another very important session on the funding aspects. So see you all soon.
Question. Can I
Please stay signed in. It's easier than locating the Zoom link again. So see you soon at 2 o'clock.